This is the second PowerPoint for photosynthesis. This will be the third video. PowerPoint 1 required 2 videos. This video is also obviously looking at the Calvin cycle, so looking at the second part of photosynthesis, as well as some cross-cutting concepts like environmental conditions and photorespiration. Again, the net overall equation for photosynthesis is this. You should know this by now, but I think what we are aware of is that the water that goes in is actually used for the light dependent reaction, and therefore we can probably assume that the carbon dioxide is used for the second part. Again, the products, we can see that oxygen is a product of the light dependent reaction, whereas the sugar is going to be a product of the light independent reaction. We also know it occurs in 2 stages. We are focusing on the second stage for this video. Also, we know that we focused on the first part of this for the first 2 videos. So the second video is talking about once I've made ATP and NADPH, that enters the Calvin cycle, which is in the stroma, the fluid inside of our grana, so around the thylakoid, and how it produces organic compounds. The Calvin cycle, or the chemical energy cycle, uses chemical energy and CO2 to produce organic compounds like glucose. The Calvin cycle occurs in the stroma of the chloroplast, which is the fluid that surrounds our little thylakoid discs. What happens here is called carbon fixation. Essentially, this is the process of assimilating carbon from a non-organic compound, so carbon dioxide, and incorporating it into an organic compound, like a sugar. So, the first step in the Calvin cycle is carbon fixation. The 3 molecules of carbon dioxide that have been absorbed from the atmosphere are joined to 3 molecules of RuBP. This is a 5-carbon sugar, and Rubisco is the enzyme that is going to join the carbons to the RuBP molecules. And so we have that here. This forms 3 molecules, which each have 6 carbons total. We have 18 carbons overall. Then we have the reduction step. These 3 6-carbon molecules are really unstable, so they split in half, forming 6 3-carbon molecules. These molecules are reduced by gaining electrons from NADPH. And so when I go from my 3 to my 6 molecules, NADPH loses its hydrogen, releasing energy in electrons. It becomes NADPH. ATP is also required for this, and it loses a phosphate to create energy. So I now have 6 3-carbon molecules, and these are known as P-gals or G3Ps. Since the Calvin cycle started with 15 carbons, 3 5-carbon molecules, and there are now 18 carbons, we have a net gain of 3 carbons. Remember, those 3 extra carbons came from the carbon dioxide. One of these extra 3-carbon molecules will exit the cycle and be used to form half of a glucose molecule. Once the Calvin cycle turns twice, those 2 molecules of G3P will combine to form 1 glucose molecule, or another organic compound. Then we need to regenerate our RuBP. Since this is the Calvin cycle, we need to end up back at the beginning. And if we remember, the beginning was 3 5-carbon molecules. The remaining stuff, so we lost 1, remember, to go make our glucose. The remaining 5 G3P molecules are rearranged to form the 3 RuBP molecules. It uses ATP, and we're back at the beginning. So if we look, carbon dioxide loses its carbons to RuBP, using Rubisco, which is an enzyme. So RuBP is my 3 molecules of 6 carbons. My NADPH is going to break those into my 6 3-carbon molecules. One of those leaves to be used to make glucose, and then the others are rearranged to make my RuBP again before it becomes my 6-carbon RuBP, so it's my 5-carbon RuBP. This is a little bit more of a complicated format of that. You're welcome to look at this as well. Quick recap. In the Calvin cycle, energy and electrons from the light reaction in ATP and NADPH and carbon dioxide from the atmosphere are used to make organic compounds. The Calvin cycle occurs in the stroma, inside the chloroplasts, and carbon dioxide, ATP, and NADPH are required. Organic compounds are produced. So this is kind of a broad overview. I will let you pause this and read through it on your own because I've kind of talked about this quite a bit at this point. And we can look at the photosynthetic equation. This is a little bit more broken down, and I would recommend writing this down as well because it kind of breaks down that general equation into the light and dark cycles. What we can see is that 3 parts of this are part of the light reaction and 2 are part of the Calvin cycle. Again, you can look at the animation if you'd like to. Some environmental factors in photosynthesis, because remember, we always need to be thinking about how does this relate to our bigger picture understanding of the world. The rate or the speed of photosynthesis can vary based on environmental conditions. This should be obvious. Light intensity, temperature, and oxygen concentration are all going to impact how quickly I can perform photosynthesis. As light intensity increases, so does the rate of photosynthesis, obviously. This is due to increased excitation of electrons, but eventually the photosystems become saturated. What that means is that there is a leveling off point, right? If we look at the rate, a horizontal line means no rate at all. No photosynthesis is occurring because my photosystems are what we call saturated. Essentially what this means is that it cannot become any more excited. Temperature affects the rate as well. The effect of temperature on the rate of photosynthesis is linked to the action of enzymes. As the temperature increases up to a certain point, the rate will increase, but then if it gets too hot, it could denature those enzymatic proteins. Basically, think back to what we learned about with enzymes. Oxygen concentration, as it increases, the rate of photosynthesis decreases. This is due to a phenomenon called photorespiration, which is a kind of dangerous phenomenon that we need to understand, especially as our understanding of the environment becomes very imperative and potentially distanced from what the government supports. Photorespiration occurs when Rubisco, or the RuBP, joins oxygen to RuBP in the first step of the Calvin cycle rather than carbon dioxide. Remember, RuBP goes from 5 carbons to 6 carbons. We can join oxygen to that instead, but that's going to become a problem. Whichever compound is present in higher concentration is what's going to be joined. So if I am oversaturated with oxygen, I will join those oxygens to the RuBP. Photorespiration prevents the synthesis of glucose and it utilizes the ATP, so we're wasting energy and we're not making any sugar. More carbon dioxide means more Rubisco joining the carbons to RuBP, which means photosynthesis occurs. More oxygen means the Rubisco joins oxygen to the RuBP and photosynthesis doesn't occur. This is primarily a problem for plants under water stress. When plants are under water stress, their stomata close to prevent water loss through transpiration. However, that also limits gas exchange. So remember, the stomata are holes on the base of the leaves, and they allow for gas exchange as well as water vapor to leave. If it gets too hot, water is going to keep leaving, and so the stomata close, but gas exchange is not going to occur. O2 is still being made through the light reaction, but it's getting trapped inside of the leaves, increasing the concentration of oxygen, and therefore CO2 can't enter the leaf, and the oxygen starts getting fixed to the ruby pea. Obviously, as the concentration of oxygen increases, CO2 decreases, and photorespiration instead of photosynthesis is favored. So, some plant species that live in hot, dry climates, where this could be a big problem, have developed mechanisms through natural selection to prevent this from occurring. They are called C4 plants and CAM plants, and you definitely need to understand this. C4 plants, which are normal plants, perform the light reaction and the Calvin cycle in the mesophyll of the leaves, so instead of elsewhere. The bundle sheath cells of C4 plants don't contain chloroplasts. Basically, C4 plants and CAM plants modify the process of C3 photosynthesis to prevent photorespiration. So, back up. These C3 plants are normal plants. Photosynthesis occurs in the mesophyll. In our C4 and our CAM plants, they're going to modify that process. They will essentially do one of two things. C4 plants will perform the Calvin cycle in a different location to avoid that buildup of oxygen. CAM plants obtain CO2 at a different time than C4 plants, allowing them to open their stomata. Both of them separate the initial fixing of CO2 from the using of CO2 in the Calvin cycle. So the C4 plants, they include corn, sugarcane, sorghum. In this process, the CO2 is transferred from the mesophyll cells to the bundle sheath cells, which are impermeable to CO2. This increases the concentration of CO2, which basically means the Calvin cycle is favored over photorespiration, and the bundle sheath cells of the C4 plants contain those chloroplasts to do that work. They use something called the Hatch-Stack pathway, and basically PEP carboxylase adds carbon dioxide to PEP, a 3-carbon compound in the mesophyll cells. Then this produces a 4-carbon compound, why it's called carbon 4 rather than carbon 3, and the 4-carbon molecule moves to the bundle sheath cells. In those bundle sheath cells, the carbon dioxide is released and the Calvin cycle begins. That Hatch-Slack pathway helps prevent photorespiration. In CAM plants, the photosynthesis includes, CAM plants that do this type of photosynthesis are succulent plants like cacti and pineapples. These plants basically open their stomata at night to obtain CO2 and release O2. This prevents them from drying out by keeping their stomata closed during the hottest part of the day and doing the gas exchange at night. When the stomata are open at night, CO2 is converted to organic acid via the C4 pathway. So technically they are C4 plants, and then it is stored overnight. During the day, when light is present, the carbon dioxide is released from the organic acid and used in the Calvin cycle to produce organic compounds. Remember, even though the CO2 is taken in at night, the Calvin cycle can't occur because the light reactions aren't occurring during the dark. So it basically is just stored until then. Both C4 and CAM plants, which are primarily found in hot, dry climates, have these evolutionary adaptations to prevent phosphorylation. C4 plants perform the Calvin cycle in the bundle sheath cells, and the CAM plants open their stomata at night and store the CO2 until the morning.